So today's session is the first for this year, for 2021-2022. And we are welcoming Dr. Dafina Angelova to this series. This is part of the Early Researchers Seminar Series that is hosted by the Cambridge Reproduction SRI. We are very grateful to the volunteer coordinators, Aideen, Grace, Gabrielle, and myself, for organizing this series, and to all of the speakers who have agreed to take part. The seminars are being held online on the third Thursday of each month at 1 p.m. and will last for an hour, except for the December 9th seminar, which will take part on the second Thursday of the month, so don't miss that. Details of forthcoming series seminars can be found on the SRI website, and Christina, the SRI coordinator, will put a link to that in the chat. The Cambridge Reproduction SRI is an interdisciplinary initiative that brings together researchers from across the University of Cambridge who have an interest in any aspect of reproduction, everything from science, technology, and medicine to arts and humanities. And we're especially keen to get some abstracts from humanities, so we can truly make this an interdisciplinary series. So please spread the word on that. Membership is open to all staff and postgraduate students and gets you access to SRI events, funding schemes, and our regular newsletters, which have research highlights, events, and opportunities in reproduction research. To join, just download the membership form from our website and send it to Christina Rosick, the SRI coordinator. And again, a link to the membership form, if you're so inclined to join, will be put in the chat. And if any postgraduate students or postdocs would like to take part in a seminar, please contact Gabrielle, and if anyone is interested in chairing a session, please do contact Grace. Again, these details will be on the SRI website. In last year's research seminar series, we had really diverse contributions, from monetization of periods to reproductive aging, and from the right to access assisted reproduction to pregnancy and birth during a pandemic. So we are very pleased to kick off this year's series with a great talk by Dr. Dafina Angelova on single-cell transcriptomic analysis and how it elucidates the cause of murine trophoblast stem cell differentiation. As a way of introduction, Dr. Angelova is a postdoc at the Department of Obstetrics and Gynaecology, where she started in 2019 after completing her PhD. She's a member of the Charnock-Jones Lab, and her research focuses on applying single-cell transcriptomics to the characterization of the placenta, as well as trophoblast cells in vitro. She is currently developing a novel microfluidics-based single-cell sequencing technology in collaboration with Nanjing Tech University and Sphere Fluidics. This will be applied to characterize the heterogeneity and complex interplay between different placental cell types, which can help uncover their role in both development and differentiation and pathology. She will speak for 20 minutes on the topic of her choice, and then we will go to Q&A. You can either, at that moment, you can either unmute yourself and ask the question, or put it in the chat and I will read it out for you. So without further ado, over to Dr. Angelova.
Thank you very much, Dafni, for the introduction, and thank you to the organizers. Right, so today I will be speaking about RNA-seq, a single-cell RNA-seq data set that we generated of differentiating mouse trophoblast stem cells, and some of the things that we have learned from this data set. So firstly, what I need to start with explaining is that mouse trophoblasts arise during the beginning of mouse development from the polar trophectoderm and extraembryonic ectoderm. And some of the first structures that form in the placenta are the ectoplacental cone and the chorion. Those structures later on during differentiation give rise to a variety of mouse trophoblast cells that form the major structures of the placenta. Those will include giant cells, syncytial trophoblasts, spongiotrophoblast, and glycogen cells. So all of those, the processes of differentiation are very important to understand. And one of the ways that we study them is that we can extract mouse trophoblast stem cells from blastocysts and maintain them in vitro. So this is a simple schematic of how they are maintained in vitro. Firstly, we need the supplementation of FGF4 and activin, which can come from a conditioned medium from feeder cells, which will maintain the stem cell identity of the mouse trophoblast stem cells. If that conditioned medium is removed, the cells will differentiate into two trajectories, one more like ectoplacental cone cells, which will then differentiate into giant cells, and another more labyrinth progenitor-like cells, which will then differentiate into syncytial trophoblast. An interesting observation is that the labyrinth progenitor-like differentiation pathway can also be induced by the inhibition of FGF4 signaling, for example, through a direct inhibitor of the MAP kinase pathway, such as a MEK kinase inhibitor. So we were interested in studying those processes, and the aims of our study included characterizing the mouse trophoblast stem cells population, uncovering the transcriptomic changes that drive the establishment of the trophoblast lineages in the early stages, and comparing alternative methods for differentiation of trophoblast stem cells. Our goal was to better understand the processes of early mouse trophoblast differentiation. So the experiment that we set up to study this was that we used the TS-EGFP cell line, that is a trophoblast stem cell line, and we differentiated the cells by two different methods. We either inhibited the MEK pathway with a specific inhibitor, or we removed the conditioned medium and FGF4 supplementation. We collected cells at different time points during differentiation, at 0 hours, so those are untreated stem cells, at 1, 4, 24, 36, or 48 hours. Then we dissociated the cells and fed the single cell suspension through Drop-Seq microfluidics that allowed us to generate barcoded libraries that allowed us to understand the transcripts that were contained in each and every cell, giving us an outlook of what the genes that are expressed in the cells are. So the initial analysis that we performed on the data set started out with trimming our reads to 50 base pairs and then aligning them to a custom transcriptome. The samples that had less than 40% alignment, we excluded and we merged our duplicates. And then we performed single-cell RNA-seq analysis with Seurat. Our initial data normalization was performed in ST-transform, and we integrated our batches in Harmony. We arrived at a final data set of more than 130,000 cells with about 1,700 reads and more than 1,000 genes per cell. Here on the right, we have a UMAP plot of our full data set integrated with Harmony, where we excluded any cells that didn't have 500 genes per cell, and we used a resolution for clustering of 0.8. Each dot on this plot resembles an individual cell, and they have been colored by cluster identity, and our clusters have been numbered by size. As you can see, this data set would be very difficult to interpret because it includes a lot of different cell types, and it also includes our two different modes of differentiation. So what we have done with downstream analyses, we extract different parts of the data set and analyze those to answer our questions. So firstly, in order to address aim 1, we wanted to find out if there is any heterogeneity within the stem cell pool, and also if there is a subpopulation of stem cells that resembles differentiating cells. What we did to answer that question was we extracted all of the T0 cells from our data set, and we found that at the most stable resolution of 0.4, those cells clustered into 4 clusters, which were transcriptionally similar to each other, as you can see by everything being quite close together on the plot. We have also scored them for cell cycle phase, where cells that are colored in blue or in red are still dividing, and cells that are colored in green have exited the cell cycle. We found that most of the cells, as you can see, are still dividing, with some cells here on the left side of our plot, higher proportion have exited the cell cycle. Finally, I have a plot here where all the cells have been colored by batch. That allows us to show that the clustering here is not driven by our sequencing batches or experimental batches, and is in fact driven by transcriptomic changes. So talking about transcriptomic changes, we wanted to find out if there are any significantly different genes between the different clusters that we identified. So we calculated differentially expressed genes with a cutoff of 0.6, which are shown here on the heat map on the left. And you can clearly see that cluster 2 is the most different as compared to all the rest of the clusters. And looking at some of the markers that we saw here, which are the most significant ones, we found markers such as CDKN1C, keratin 8, and keratin 18. Those ones are linked to stem cell differentiation. So we were interested to see whether the top markers that we found in our analysis would correspond to the expression patterns in later time point cells, which would be more differentiated. So here I have an illustration of that on the right. Here we have our T0 data set with expression levels colored for every gene on the top 4 differentially expressed genes. And here on the bottom, we have extracted the same data from the full MEK inhibition data set, where you can see that the island of cells here on the left contains the early time point cells, and the island on the right contains the later time point cells. So you can see that expression over time increases for all of those genes, indicating that cluster 2 is indeed more similar to our later time points and is a driver of heterogeneity. So next we wanted to find out what the data looks like during the whole time course. And to answer aim 2, we wanted to find out what are the marker genes that the differentiating cells express, what are the pathways that are enriched, can we identify any differentiation trajectories, and what are the regulons that are active during this differentiation. So in order to simplify things for this presentation, I will just be talking about the inhibition data set, but a lot of the findings that we have are corresponding for both data sets. So here, let's talk about what our inhibition data set looks like to start with. We have here UMaps of the data set integrated with Harmony again, where we have 500 genes per cell cutoff and a resolution of 0.8. We have our clusters here, colored by cluster identity and numbered by size. Here we have our cells scored for cell cycle stage, and you can see that the cells that belong to the early time points here on the left are still dividing, while they're not dividing quite as much here on the right. Here we have our cells colored by time point, and an interesting observation is that we can see that our T0 cells are mostly all clustering in cluster 9, which we have identified later on as our T0 cluster. Finally, we performed pseudotime analysis, where we found that the main trajectories for differentiation go from our T0 cluster to 2 latest points in pseudotime, one going up here to cluster 3 and one going down here on the right to cluster 4. So in order to find out what are the cell types that we're looking at and what are potential differentiation trajectories, one way to think about it would be to look at what kind of marker genes are the different clusters expressing. And here we have some marker genes that I have selected from literature that are shown as a dot plot, where the size of the dot shows the percentage of cells from the cluster that express the gene. And the more purple the dot is, the higher the average expression. We have our clusters here sorted by time point with the package tempora, where our earliest clusters, you see 9 here on the bottom, are on the bottom, and the latest ones are on the top. So we have 3 markers for each main differentiation trajectory and for stem cells. You can see that our stem cell markers, IOMI, ZEL5, and SOX2, are still expressed in the early time points and reduce expression over time. our ectoplacental cone markers are increasing in expression over time, and so do our labyrinth progenitor markers, with cluster 3 having the highest levels of expression for ectoplacental cone markers together with cluster 0, while cluster 4 having the highest levels of expression for labyrinth progenitor markers. So this gave us an indication for what we might be looking at in the future, but it's not enough to identify what is happening in the data set. So the next thing that we looked at was what are the enriched pathways that occur during the course of differentiation in the inhibition data set. So we performed pathway enrichment analysis of differentially expressed genes of the T0 cluster 9 versus every other cluster in the data set. We performed this using the enrich-R package with the database of wiki pathways, is what I'm showing here on the below heat map. So from what you can see, the greenness of the square shown here represents the p-value, and the most significant terms that we see here includes terms such as cytoplasmic ribosomal proteins, mRNA processing, translation factors, electron transport chain, oxidative phosphorylation. So a lot of biosynthetic energy metabolism pathways. So as you can see, a lot of those are very highly enriched, but we don't know whether they are increasing or decreasing. And we've come up with a way to easily illustrate whether the pathways are increasing or decreasing over time in each cluster by giving a score for each pathway depending on whether the genes in the pathway are elevated or decreased as compared to the average level for the entire data set. And you can see here on the heat map, the pink indicates significantly increased genes and blue indicates a number of significantly decreased genes. And we have our clusters sorted here by time point again. So you can observe that in the beginning time points, we have higher levels of activity in these 4 pathways that are the most significant, while going towards the right in the later time points, they reduce in activity. This suggests that the metabolism, energy metabolism, and biosynthesis pathways are decreasing in activity over the course of differentiation. Another way to look at the data was to identify differentiation trajectories, whether we can identify the ones that are have been predicted by previous experiments and how far do they go. So we have our cells here scored for pseudotime, and we have managed to track 2 trajectories that go from cluster 9, which is the T0 cluster, all the way to cluster 4, which I have called the labyrinth progenitor trajectory, and from cluster 9 to cluster 3, which I have called the ectoplacental cone trajectory. These trajectories are linked to cluster 4 and cluster 3. Monaco has allowed us to calculate gene expression modules that show genes that are co-expressed in the cells that belong to those regions. Looking at those gene expression modules, we wanted to see how the genes that are most significantly associated with them are going to change over the trajectory in pseudotime. So here we have done a bit of an illustration of how they change. We have a selection of the most significantly enriched genes from those gene expression modules with the highest Monaco scores, and also some widely known markers. For example, we have GCM1 and GJB2 for a labyrinth progenitor identity. And you can see that both of those are increasing over time, with GCM1 increasing at the very last step of the differentiation trajectory that we have recorded. We have also identified a number of other genes, which are more novel markers for this early level of differentiation, but some of them have been identified in a mouse labyrinth. We've done the same analysis for the ectoplacental cone trajectory, and there we found that the known markers of ACL2, FILDA2, and PLAC1 are increasing in expression over time quite significantly. And we've also found some other ones, including FGFBP1, HMGM5, and UGP2 that are also increasing in expression over time. Another question that needed to be answered about this data set is what is the activity of transcription factor regulons over the course of differentiation after MEK inhibition? We calculated those using the package scenic, And we selected the top 10 regulons that had the highest and lowest activity in the labyrinth progenitor and ectoplacental cone clusters to display here on a heat map, as well as the top 10 regulons that are highest in the T0 cluster 9. So you can identify which regulons are most highly active in the T0 cells by looking at the activity levels of L5 and EOMEs, which are stem cell markers. You can see that they're clustering together quite nicely with a big group of other markers here. While on the other hand, the cells, the clusters that have the highest activity for GCM1 and ASCL2 have been shown here together with another set of transcription factors that they're clustering together with. Another way to illustrate this analysis would be to take the scores for transcription factor activity or regulon activity calculated by IUCell and to score each cell on a UMAP that we've seen before. So here we've taken the three most prominent transcription factors that we have identified, and we have scored them for EOME's GCM1 and ASCL2 activity. So you can see that EOME's activity is the highest in our early time point or T0 cells and decreases over time, while the activity of GCM1 increases gradually until it reaches a peak in the labyrinth progenitor clusters here, while the ASCL2 activity increases until it reaches a peak in the ectoplacental cone progenitor. So that fits nicely with our previous interpretation. But another thing that we wanted to find out from this analysis is whether we can identify any a bit more novel regulons that are going to be associated with a differentiation trajectory. In this data set, we've managed to identify two regulons, PHF8 and E2F8, which are highly associated with the labyrinth progenitor differentiation trajectory. And we looked at those with a bit more detail. So here we have on the left the same plot that I showed before. And here we have scoring for E2F8 on every cell of the data set. You can see that the activity level of E2F8 increases gradually over time and reaches a peak in the labyrinth progenitor cluster. And a similar situation is with PHF8, where activity increases over time, although the baseline activity for this transcription factor is higher and reaches a peak in the labyrinth progenitor cluster. We were also interested in looking at what are the targets for those transcription factors. And interestingly, we found that some of the high confidence targets for E2F8 include 4 out of the top 10 labyrinth progenitor gene expression module. And also for PHF8, we found that its high confidence targets include PHF8 itself, E2F8, and GCM1. So this analysis would suggest that those 3 transcription factors work together to establish a labyrinth progenitor trajectory of differentiation. So that was quite an interesting finding, but there was more to look at in this data set. The next thing that we wanted to look at a little bit was how we can compare the alternative methods of differentiation. 2 questions that we were interested in is whether the methods result in distinct transcriptomic changes and whether one trajectory is favored over another in differentiation. So you remember that we differentiated ourselves with 2 methods, MEK inhibition and conditioned medium removal. So we have split the 2, and here on the top we have plots that you have seen before for the inhibition data set. Well, on the bottom, we have the same plots with the same parameters for the removal data set. So there are some things that we can see even just from looking at those plots. Firstly, you can see that there is a more gradual transition between the early and later time point cells in the removal data set as compared to the inhibition, where in inhibition they are very strictly segregated between each other. Additionally, the exit from the cell cycle appears to occur a bit slower in the removal data set. And finally, it appears that pseudotime progresses a bit slower in the removal data set as well. So those observations would suggest that transcriptomic changes are occurring slower in the removal data set. Another way to look at that was to look at markers that are significantly higher or lower between the two data sets. So we have selected the top 8 that are consistently higher or consistently lower in inhibition or removal. And we have plotted them here over time with these lines. And you can see just from looking at the lines that whenever a marker, when you're looking at them just from far away, whenever a marker is changing, it is always changing faster in the inhibition data set than it is changing in the removal data set. There is one exception in the top 8 markers to this rule, and that is FILDA2. FILDA2 is a gene that is linked to an ectoplacental cone trajectory, and it is elevated in the removal data set, which I found to be interesting because it suggests that there might be a preference for ectoplacental cone trajectory in the removal data set. So we wanted to look at these two data sets in comparison in another way. So we thought a good way to compare them would be to look at regulon activity again. Here we have all of the regulons that were found to be active in the inhibition and removal data sets. The first 2 heat maps here show the regulons that both inhibition and removal have in common. And you can see that the vast majority of the ones that were found in the data sets, they have in common. I'd like to direct your attention to ASCL2 in particular, that is present in both data sets, and it appears to be present with similar patterns of activity over time. So in contrast, here we have regulons that are unique either to inhibition or removal. An interesting observation here is that GCM1, a main marker for a labyrinth progenitor identity, is only present in the inhibition dataset. That would suggest that it is below the detection threshold for the removal dataset, and also suggests that it's possible that this is because GCM1 has lower activity. So these are scores for the activity of the transcription factor. Another way to look at these questions for ASCL2 and GCM1 as markers for ectoplacental cone or labyrinth progenitor would be to look at the expression of the transcription factors themselves. So we plotted those over time as well. And we found that in the inhibition dataset, GCM1 peaks earlier and more consistently, while in the removal dataset, that is true for ASCL2. So all of those bits of information together with the fact that the proportion of labyrinth progenitor cells in the inhibition dataset is much higher than the proportion of labyrinth progenitor cells in the removal dataset, it would suggest that this differentiation trajectory is favored in our trophoblast stem cells after MEK inhibition. So this was an interesting observation as well. And with this, I would like to finish and summarize everything that I've talked about today. Firstly, we learned that heterogeneity is present in trophoblast stem cells with one cluster of cells resembling differentiating cells. We also found, looking at the entire time course, that cell division and energy metabolism pathways decrease in activity over the time course, and that the 2 main trajectories we identified were labyrinth progenitors and ectoplacental cone. We also identified the 2 regulons E2F8 and PHF8 are highly active in our labyrinth progenitor trajectory. And finally, we found that differentiation appears to progress faster after MEK inhibition, and also it appears that the labyrinth progenitor trajectory is favored after MEK inhibition. So this is just a subset of the analyses that we have done on this data set. And there is also many validation experiments that can come forward from these analyses. And I'd just like to speak a little bit about the first experiment that we are working on right now. Just a little bit of validation work. We wanted to start with trophoblast stem cells that we have differentiated for 48 hours, so at the end of our time course. And we would like to label them for some of the novel markers that we have identified for our 2 differentiation trajectories. We would like to separate those cell populations, plate them out separately, and try and differentiate them for a further 4 days, at which point, hopefully they will acquire the markers for syncytial trophoblast or giant cells that are more established, and we will be able to stain for them and identify whether our predictions were correct. And with this, I would like to finish. I would like to thank everybody that has participated in this project. In particular, I'd like to thank my supervisor, Steve, Wendy Bacon, who generated the data set, Malvina and Russell, who were with the CTR and did the data analysis. And finally, I'd like to thank Miriam Himberger, who has helped us immensely with the cell culture, some of the data analysis, and now with the validation experiments. It's been great to work with all of them, and I've learned a lot from all of them over the last 2 years. Thank you very much.